Alright, so you guessed right, since you're seeing that the Henson's node is a structure directly opposite to the region identified as P a while ago. This Henson's node actually marks the anterior portion of our animal. Now, now, if we're going to make a cross section of any of these portions, we will be able to appreciate under the microscope a segment or a structure or an image similar to this. So, if we take a look at this cross section on the upper panel, you can still appreciate the presence of the area opaca and the area pellucida and the canal that is actually produced during the gastrulation stage. Now, the solid red arrows depict the direction of cell movement from the surface of the growing embryoblast towards the inner portion of the animal. Alright. More so, um, here on the lower cross section, we can identify three germ layers that have been established or that is supposed to be established after gastrulation actually finishes. The outermost layer, which will not be in touch with the yolk, becomes ectoderm. Then the layer that is actually on the bottom portion of this cross section, we expect that this layer touches the yolk. Let's say we imagine that the yolk is found in this region here where I'm circling my arrow. So the layer is known as the endoderm, and the space between the ecto and the endoderm is filled by cells of the mesodermal layer. Now, let's take a look at further structures that develop in later hours of incubation. So, this whole structure here is said to be the primitive streak still. You can see the primitive ridges, these two ridges here. Then the lightly staining structure in the middle looks like a canal is your primitive groove. Um, Now, another structure is a proamnion. This proamnion part here is actually not going to be a part of the amnion, but it's defined as collective compaction or compaction of cells, aggregates of cells on on the more cephalic region, more anterior region of the growing chicken embryo. This area is free from mesoderm. No, it 18 hours late 18 hours. You can see a smaller primitive streak compared to the one a while ago. So in this stage, we can see that an area known as the head process is beginning to manifest. So later on, this head process will have dramatic changes, like particularly in the 24-hour stage. So let's take a look at the 24-hour stage. 24-hour stage of chicken embryo, you can appreciate a lot of changes on the growing animal. Let's first examine the head part, the head process part, where more anterior regions are expected to be found. Okay, so here we can see uh, the ridges a while ago. No, this not the ridges, no, but let's see. Oh, oh, okay, so here the ridges a while ago, characteristic of the primitive streak, now becoming smaller as the hours of incubation pass by. Right, the anterior region of the animal is showcasing more head-like formations, right? Like anterior neurophore. No, this anterior neurophore is actually the unclosed or uncovered area of the neural tube. No? Also, we can see that neurulation actually begin this part. You can see neural folds of the animal. This one, darkly stained structure, neural folds that later on will close to form one continuous long tube known as the neural tube. Okay, you still have some parts, similar parts, like some parts that we've